Hello friends, this video on Biotechnology Principles Part 18 is brought to you by examfear.com. No more fear from exam. Now the next step, which is going to be new now, because till here we have partially discussed previously also, but from here it is going to be new. So here we are going to talk about the amplification of gene of interest. Now recombinant DNA also is formed. Now before the recombinant DNA is introduced into the host cell, it is important to amplify the gene of interest, because as I mentioned before also that the gene of interest is crucial to us. The entire focus is on the gene of interest, and just in and around the gene of interest to talk about the entire concept of genetic engineering. So it is very important to amplify the gene of interest, and this process is performed in laboratory conditions. So what happens in this process? This amplification is done with the help of a technique called Pcr technique, which is polymerase chain reaction technique. So Pcr is polymerase chain reaction. Now we will understand the entire reaction. So this is a technique to produce multiple copies of a piece of DNA or a sequence of DNA. So when we say gene of interest, what is gene of interest? It is nothing but a particular sequence of DNA. A particular sequence of DNA forms a gene. So our desired gene is the gene of interest. So now if we want to amplify that gene, what we mean is we want to create multiple copies of that gene. Now, this Pcr technique is a process which can be carried out in laboratory without having any dependence on any bacteria or any other living organism. So that is the advantage of this technique, that it is independent of any other organism, and you can perform this in a laboratory and you can create multiple copies of a piece of DNA. Now this technique was developed for the first time in around 1983 by Carey Mullis, and that is why he is famous for amplifying the gene of interest for the first time using the Pcr technique. So now we will try to understand how Pcr technique works. So now we will talk about polymerase chain reaction. So let us look at the various components which are needed for this polymerase chain reaction. So the first component that would be needed is a DNA template. Now by now I am sure all of you know what are these terminologies. I mean, what is a DNA template? Now first of all, this DNA is made up of two strands, right? One you can see, one is the red strand, the other one is the blue strand. So if one of these strands is running from 5' to 3' direction, then the other strand is going to be anti-parallel. That is, it will be running from 3' to 5' direction. So that is how the double-stranded structure of DNA is. Now what happens here is, when we want to multiply or when we want to create a lot of replicas of this DNA, what do? We need a DNA template. That is, if you have one strand of DNA, the other strand can be created, because what is that one strand? That one strand is basically the sequence of bases like A, G, T, C and so on. So the other strand can be created by creating the complementary bases of each of these, and that is how you can create the other strand. So for creating this new strand, you need the template strand. So that is what, in polymerase chain reaction also you need the DNA template. You need DNA polymerase enzyme. This plays the most important role, and that is why this reaction is called DNA polymerase. This is called polymerase chain reaction after the name of the enzyme DNA polymerase. And the third thing that is required is two sets of primers. Now what are these primers? Okay, we will see that. So let us try to understand each of these. So what is a DNA template? So DNA template contains the target DNA region to be amplified. Now first of all, we have to define that which portion of DNA needs to be amplified. So let us suppose in this entire strand of DNA, if I say that only this much portion needs to be amplified. So what does that mean? How exactly the DNA replication take place? If you all remember the process of DNA replication, so if this is a DNA strand, double-stranded DNA, so this gets separated, the two strands separate out, and then this DNA polymerase enzyme comes and it starts reading each of these strands and it creates the complementary strand, and that is how from one DNA, so this was just one DNA, from one DNA you get two DNA like this. So that is how DNA replicates. So here also, first of all, we should know the DNA region which needs to be amplified. So that region of DNA acts as the template. Next is the polymerase enzyme, because this is the enzyme which has the ability to create DNA strands. New strands can be created from nucleotides. How? Because this enzyme can continuously keep on adding one nucleotide at the end of an existing strand, and that is how it can create the entire DNA strand. So what does it do? It reads the template strand and adds complementary nucleotides. So let us suppose this is the template strand. So if this is A, so the DNA polymerase will add T here. If this is G, DNA polymerase will add C here. So that is how it can create a new strand, because it has the ability to read the existing template strand and add complementary nucleotides accordingly. Extends the primed segments, adding free nucleotides, and it has also the ability to keep on adding new nucleotides, and multiple nucleotides together form the polynucleotide. So DNA polymerase does the main thing, that is the new strand creation is done by DNA polymerase enzyme. So what are primers? Now primers are not one single nucleotide but oligonucleotide, that means few nucleotides joined together, that is called oligonucleotides. Complementary to 3' ends of both DNA strands. Now what happens is, let us try to understand this coiled structure of DNA in a simple way. Let us suppose this is one strand of DNA, this is another strand of DNA. Let us suppose this is the 5' to 3' strand of DNA, and this one is the 3' to 5' strand of DNA. So just to keep things simple, I am not coiling it. So let us suppose this is 5', 3', that is the blue strand, and this one is the red strand. Right? Now what happens is, this is one single strand. So this, based on this, a new strand gets created. Now for the creation to start, what happens is, small oligonucleotide is present towards the 3' end of both the strand. So this is the 3' end. So towards the 3' end, there is a small oligonucleotide present. That is, maybe 2-3 nucleotides are present which are complementary to this strand. So here also this primer is complementary to this strand. So this is called primer. Why it is called primer? Primer is derived from the word primary, because it initiates the process. So it is a primary thing. So that is why it is called primer. Now on this primer, then DNA polymerase comes into picture, and then it extends this primer. So on this primer only, it keep on extending it by adding the complementary nucleotides, because DNA polymerase can read this existing strand and it can also add nucleotides to an existing strand. So existing thing is primer. So DNA polymerase will keep on adding more and more nucleotides to the primer by adding complementary nucleotides to the existing strand. Similarly, this case also, it will keep on adding more nucleotides to the primer. So as a result, here you get another strand which runs from 3' to 5'. Here you get another strand which is running from 5' to 3'. So if you look at this structure in itself is a DNA. So you get one DNA here and you get one DNA here. So that is how you get two DNAs from one DNA. So this is how polymerase chain reaction will take place. Again, from each of these you get two. Again, from this you get two. And that is how the chain reaction keeps on expanding. So you basically start with one and you end up having many. So the concept of polymerase chain reaction goes somewhat like this. Thank you. Please visit www.examfear.com to watch more educational videos with a better experience. Please do not forget to like and subscribe to our YouTube channel for latest updates. Thank you once again.